biology and lots of different subject matter expertise. So our aim today comes from the fact that, as a lot of teams have said, NF2 is a really rare cancer and there's very little data to understand it, to understand how to treat it. So our goal was to look at cancers that are known and sequenced and well characterized and ask, can we find a subset of these known cancers that are biologically similar to our NF2 patient here? And can we use such a subset to suggest treatments or particular pathways that might be relevant to our NF2 patient? So our approach, for our data set, we had the VCF of the mutations that are present in Onno's tumor genome. And for existing cancers, we took TCGA data sets showing the mutations found in patients from the Cancer Genome Atlas. Since we had limited time, we took only 576 brain cancers. And we had two strategies for clustering these. The first is based on mutational signatures. I'm happy to discuss in more detail, but in a limited time, point mutations can be divided into 96 categories based on the base that is mutated, what base it's mutated into, and the base before that and after that. So basically, what's the mutation and what sequence context does it happen in? And the biological processes that cause mutations, basically mutagens, damage DNA repair pathways, enzymes that go and cause damage to the genes, cause specific patterns of mutations across these 96 categories. And these are mutational signatures. So this is an example of a biological process that mutates 4 out of 96 categories. So the first parameter which we used for clustering was mutational signatures present in the cancers, which tells us what kind of processes are acting on these patients' DNA. The second was the genes that are mutated in these cancers, and specifically the GO-terms, the GO-terms of those mutated genes, which indicates the biological function that is carried out by mutated genes. So basically, what biological processes are damaged in these cancers. So to do this, we took our data sets, counted trinucleotide mutations in each genome, and extracted known mutational signatures using this R package. We also identified mutated genes in each cancer genome and identified the biological function Go annotation of each gene and counted the number of genes that are mutated in each Go term. And then we used dimensionality reduction, PCA, and clustered all these cancers using k-means. So I'll briefly present our results. We filtered Onno's VCF to identify 9,600 roughly somatic mutations in his tumor. These are high-quality variants with more than 15 reads supporting the mutation and are not present in his normal tissue. We identified the mutational pattern across the 96 types of point mutations, and we identified 229 protein coding mutations in 43 genes. So these are our clustering results. Based on the mutation signatures, we identified 7 subtypes of brain cancer. And Onno's tumor is this X over here, which maps to the least populated cluster. This cluster is mostly glioblastomas and is characterized by specific mutational signatures, including what is known as signature 3. This is signature 3. It's normally found in another very well-studied type of inherited cancer, which is the BRCA-positive breast cancers, and as well as other cancers. Basically, the signature is found in cancers that are defective in the homologous repair DNA repair pathway, and these cancers are specifically treated by a class of drugs called PARP inhibitors. So that's an example of how we can use this strategy to identify similar cancers and identify a treatment mechanism for a patient. We then went on to the functional clustering. So this is based on what biological pathways are damaged in these cancers. And on this basis, we identified 5 subtypes of brain cancer. And this was very interesting to us because Onno's tumor is a total outlier over here. And so functionally, our NF2 patient is very, very different from all known categories of brain cancer. So our future directions. We definitely want to extend this analysis, particularly because we see that NF2 is very different from known brain cancers. What if we go ahead and look at all types of cancers, breast cancer, lung cancer, everything we know, and try to find a cluster that's similar. Will it be similar to something unexpected? We would also love to do this using other data types, including gene expression and things that could give us a better functional characteristic of the pathways that are affected in these tumors. We want to deepen our analysis, and if we find, based on various parameters, if we find that our patient is similar to some known group of tumors, we want to look very carefully at what pathways are affected in those patients and how we can use those to identify specific mutations that we can target. And basically, we gave an example of how we can use this strategy to predict treatment, and if we continue to broaden and deepen this analysis, hopefully we'll end up with a list of possible targets. Thank you.
Wow, that was really impressive. Thank you guys very much. I cannot wait. Is all the code on GitHub? Awesome. I cannot wait to dig into it. I'm sure I speak for a lot of people who love to take a look. All right. Thank you guys again. You guys are amazing. Snip Pixel. Hey guys. Oh yeah, here you drive. We are very, very close.
All right, so we're Team Snip Pixel, and it's our honor to be here and work with everybody, you know, great guys, and I know everybody's excited for the celebration afterwards, so I was trying to be as quick as possible. So, and it's also our great teammates, and we're lacking two here, and we also missing a beautiful lady, maybe make us less attractive, but anyways, so. So yeah, the idea behind this Snip Pixel is to find the right pixel in the genome. In this case, we're working on Onno's genome, and we're specifically designed a strategy to find the NF2 mutations. So, but we're trying to keep all the variations secret, not to leave too much of a secrecy. But anyways, so we first annotated the SNPs. Then the approaches we took is we predict the effects of the variants on the genome based on the amino acid changes, and also based on the regulatory elements, which the variations are not in the coding exon, where it could be in the introgenic region, or like upstream 3', 5' UTRs that also affecting the expression of the gene. And also, we specifically designed an approach. We actually look backwards from the very basics about how NF2 works, because it's a rare disease, and we screen the candidate variants based on inherited and sporadic mutations. And also we monitor from the variant frequency in the populations to give us more prioritize the mutations. So this is a basic idea of the how we from the VCF files we get. We look at the different samples. So this normal blood sample is actually on Onno's normal tissue, where we assume that it carries a one inherent homozygous mutation, which could be one of the cause of mutations of NF2. And in tumor tissue, that the homozygous mutation that's actually making the NF2 tumor cancer tissue to grow. So for a normal non-NF2 population, we imagine that most people will carry a uppercase N, so that's a homozygous wild type. And so quick demo here. Here. So we first we combined all the VCF files we combined all the samples into one VCF file where it lists all the samples and its conditions. And the specific strategy we designed for NF2 is as I mentioned. So we took it to evaluation of the pathways, and somehow the picture is too big to load, but we can go to the. We link the pathway enrichment to Reactome, which gives us a confidence of all the pathways that affects the mutation. So we see here what is really surprising for us to see. So you see here the most enriched pathway are actually the first top two, actually. They are all located in neural systems, which is, a kind of, surprising and exciting to see. That. So diving into the detail of this RSK activation pathway, we see it's partially related to EGFR pathway, where we found several intron variants and also some exon sporadic mutations on chromosome X. There are also Nature and Science papers indicating the link of chromosome X and cancer, because especially in the case of male, it has one copy of chromosome X can lead to say the sporadic mutation on chromosome X could have a higher potential of cancer development. And we also noticed these. This is just one. Might be too small to see, but this is just one example of the mutations that we found. It's about 3%, right? 0.3. Yeah, less than 3% of that mutation. While Onno carries two, rare mutations. So that's, you're very unique. So yeah, but that's our one of the proposed pathways here is that we should give the focus on this pathway and potentially find some, maybe apply deep camming there to find drugs in this pathway. And also, since we're from Minnesota and Recombinetics, we're a gene editing, company that build pig models using CRISPRs to model different diseases. And we also currently have an NF2 project going on for that. So, yeah, thanks.
That was amazing. Thank you guys so much. Please go for a picture. The earlier comments notwithstanding. Yeah, do you have Cosmos there? Yeah, or I can talk about Cosmos. Oh, yeah. Yeah. So I think everybody met Cosmo. Cosmo was a mentor and also working on things. And the thing he said to me most frequently was, I found another variant in Onno that I think is related to disease. So let me show you some of them. So he's really excited about a tool called Mendel MD, which is an open source web server that runs lots of stuff. He found 30 genes with high impact variants, and he was particularly excited about a few of them. And I think that's really exciting. One of them He has stop insertions that he found in Onno. It's in several NF2 patients. So that's a pretty obvious mutation. So he built a tool called massdb.org, and he could visualize the structure and location of that. What other variants connect? Back to the hit list. One of the judges proved that ANAPC1 is linked. So thank you for that, Margot. And he was very excited about this one too, where two isoforms of 3 for USB 17L with stop gain. Oh, okay. Anybody know what a stop gain is? I'm not quite sure what a stop gain is. Oh, it's a gain of a stop. Okay, gotcha. Anyway, thank you. I'm really glad he's here. Anyway, he also found dump3, yet another thing, and clearly these things really sort of contribute to parts of the network. Oh my gosh, there are more of these. Yeah, cryptokinase, there's a frame shift mutation. It inhibits PAC1, which is also involved with NF2. And here's the last one. A gain of stop. It's right next door to NF2. Oh yeah, he was telling me about this last night. It's right next door, which is really interesting. And I encourage him to think about deletions. And there are also deletions that are known. And so here you can see what happens when you have a deletion, which thankfully none of this stuff. And so, yeah, 30 critical impact, and a lot of them are already known. So that's actually really cool. And that's Cosmo's presentation. So, Cosmo had to leave suddenly to do something really, really important. Do you want to say something about that?
Well, he called it the perfect storm.
Yes. So lots of things that have little effects on a protein pathway all aggregated together. So really very interesting. This is Bill. Bill was here Friday. I'm gonna let Onno present this.
Oh, okay. Well, I haven't seen it, so I know Bill. Bill actually is another patient, not an NF2, but another cancer patient, and looking for also, treatment options. So I think it's like he found some things that might be also related, but I haven't seen it. So I'm seeing this for the first time too. The spurs have been studied. Yeah, I think so. These are all the drugs that have been studied for NF2. And yeah, they look at the signaling pathways.
We've seen RTK in other presentations. Oh, I was just going to say we've seen RTK in other presentations. So he thinks Avastin, surprisingly, may have, there may be some promise to Avastin. I've never heard of Avastin being used for this. You guys have?
Most common with apps.
Yeah.
Oh. Number one treatment.
Okay.
Known FDA.
What's that?
Known FDA approved for NF2.
Oh, okay. It's FDA approved for NF2?
No.
No, not.
Oh, it's non-FDA.
It's not.
Got it. Okay. I think that's it. Big hand for Bill. What's that? Oh, pick some teams that are sitting here. I think, has anybody that wants to present not presented?
Yeah.
We skipped you.
Yeah, I know.
Oh no, had a reason.
That's my randomness.
This is going to be cool.
Hello everyone. So as you can see, we have a girl-only team. So we have Len, who's a statistician, Raquel, who's a computer scientist, and my name is Joanna, and I'm a computational biologist. And our team is called Mutation. So the overall goal of our project was to identify new potential drugs for schwannomas. So as a starting point, we used the results from this study that was published in 2002, and it happens to come from the lab that I work in currently. So in this study, this was the very first study that used microarrays for gene expression profiling of soft tissue sarcomas. And so soft tissue sarcomas are tumors that are derived from different types of connective tissues. And this study happened to include also 2 samples from patients with schwannomas. So based on this gene expression profiling study, there were over 1,700 genes that were identified to be either up or down regulated compared to those other types of tumors. So we used this list of the genes as the starting point. And later we used Onno's tumor data, tumor compared to normal, to search for potential actionable mutations in those genes that were shown before to be dysregulated in schwannomas. So we looked for variants present in exons and also regulatory regions of those genes, and we found several deleterious exonic variants. So by deleterious, we mean that they may affect the protein function. We did some additional screening, like we checked for the mappability of the regions where those variants were identified to see these are not just like technical artifacts of the sequencing. And and 2 of those genes were shown before to be overexpressed in schwannomas, and 1 of those genes was downregulated. And then we searched NCBI resources to see if there are any drugs that are known to target any of those genes. And actually, there are 2 drugs that are known to inhibit one of the proteins. So, and what is very interesting is that those drugs were already shown to be active in this cell line that is called SVAI, which is actually a model used in research for studying neuronal function and differentiation. So, very similar type of cells that, exactly the same type of cells that schwannomas are derived from. So this is just a print screen from NCBI showing PubChem information summary about those. Yeah. So yeah, you can see that it indicates that there are some compounds that may be active against this target, and one of those compounds have a very fancy name, Shambo 38752, but apparently it's registered to potentially treat nervous system disorders, and it may possibly be also an anti-cancer drug. And the second drug is inositol triphosphate, and it has been shown that it's implicated in sphingolipid pathway, so it's also associated with the function of neuronal cells. And yeah, that's the summary of our findings. So yeah, we'd like to thank all of the organizers for putting this event together. It's very exciting to have the opportunity to participate in this hackathon. Thank you very much.
That was great, and also the shortest presentation we've had so far. Thank you. That's you guys? No, that's somebody else. But you can. Yeah. Barely understand it? They're right there. Is barely understand it. Yeah. Yeah, they're right there. They're coming up. Yeah, yeah.
Hi, so we're taking barely understand. And so we trained a machine learning model, well, a couple things. One was look at various variants and features that might be used in machine learning models to predict epitope finding. And the other was to sort of characterize potential epitopes for treatment and immunotherapy.
My name is Rui from Genomics. I was working on identifying specific antigens from genome sequence data. So here's some background on how we can identify this kind of antigen for immunotherapy. So you can see green, kind green stuff that is antigen. This contains some tumor mutation called new antigen. So basically, we have tumor model data, so which is perfect for our pipeline. So we can compare tumor model and find some missense mutation and map into the transcriptome and chop to the peptide and predict the peptide interaction between peptide and actually molecules. So that's a pipeline we created about 1.5 years ago. So basically, as I said, it's compare tumor model data and fill out a set of sense mutation and map peptide using NANHCN to predict affinities. So because we don't have almost actually genotyping data, so we cannot do any more for prediction. But we are using some common view like actually BA1, which is quite common in computation. So using this iterate view, we go through the pipeline, and here's a part of the result from the pipeline, which is a list of missense mutation count in each gene. So the second gene is the most interesting part is MDGFP1. So we have 1 missense variants in this gene, and what we compare to the set. And here's one A change to C, the gene of A or C that include different peptides. One's changing amino acid from F to C. So if you predict the binding affinity, you see IC15. So the tumors are pretty high. Tumors are low, which means they have a strong bind. Normal is high. So which means the normal tissue will never present this set of peptide into cell surface. And T cell will never educated by this set of peptide. So once the tumor cell produce this peptide presented in the cell surface, which means T cell never seen this before. They'll think it's a non-cell, so they are attacked as well. So this is tumor specific target we're trying to identify. Well, once we come here and need some guys to, I asked a question. So this is a critical part for prediction of binding affinity. But actually, genes highly polymorphic. So for some common view, we have a lot of training data set. We train your neural network. But for some rare view, it's not that good. So I asked the question, can we do some improvement for this tool? So that is these 2 days work. Such a thing. So I'll give my
This is supposed to be proof of concept, but due to lots of time constraints, it's going to be of concept. Yeah, ignore the results. I mean, it does do better. The technique I'll talk about shortly, but this is high variance and whatnot going on. Long story short, we used a one-hot encoding on these binding sequences within the motif, and we took that data, and then we also tried to incorporate some sort of structural data. And so to do that, we used a technique from algebraic topology called precision homology. And the idea there is that you're math savvy, you're understanding the homology of the underlying manifold. I mean, no sense whatsoever. You're trying to look at the shape of your data and use some sort of feature discrimination based on that. So you can sort of run this black box, and then you'll get some sort of distance metric. And we presented that to the network as well. it's ready to go. We just need time to run it. So I'd be interested to see what happens, and I guess that'd be like a good result. Basically, the idea behind this data set, or the method, excuse me, is that you often have many different types of features that you want to look at, but in a variety of scientific fields, like in neuroscience, you'll just threshold and say, oh, this is like biologically possible, or give some reasoning behind it. Although that may not make any sense. So by integrating sort of features across a variety of scales and then looking at a mathematical object that encodes all that information, you might actually get significant performance boosts, and that's been shown in a variety of fields, including archaeology, networks, neuroscience, cosmology, and a variety of other areas. So we're interested to see if this works. Basically, we, as far as the pipeline, did got crystal structures, trying to create, maybe it's not the native form of the protein, but then we used BioPython to take alpha carbon positionings and then look at a distance matrix of all the values. You can then run this through the topological feature extraction and do bottleneck distance, which is a various metric that this thing's numerically stable under, and then feed that to your network. So that's like ready to go. Not sure what's going to happen. Be interesting to see.
Before we have that potentially better method to work, we did some standard comparison of existing methods for predicting the binding affinity between HLA and peptide. So these two are the most state-of-the-art methods out there. They both use machine learning. They both use neural networks. One uses deep learning, one uses just one hidden layer. So you can see that for the predictions that we throw in there, they tend to cluster. So there's quite some uniformity within the low end, high end. The low end means the two molecules are binding very tightly, and the high end means they're quite loosely. So what we are really interested in is this lower left-hand corner, where the two things bind pretty tightly. That's potential targets for this immunotherapy. So we basically zoomed in and took this tight binding peptides, and we realized that there's a lot of overlap between these predictions. So even if our methods don't work, the platforms that we have developed, which is our first contribution here, actually allow us to propose some candidate peptides for immunotherapy. So some other contributions, we verified some comparisons for the methods, and we realized that these methods are potentially good enough for predicting some candidate strong binders. And lastly, we want to improve beyond standard prediction methods.
That was fantastic. Thank you guys very much. I mean, I think that's very exciting. Go get a picture of your team. Who are we still waiting on?
Can you give the name to this man?
All right, so we have a couple more teams. If you're watching this on YouTube and we haven't gotten your remote team, please message the Hackathons channel now. But without further ado, Team Base Pair. There's going to be some cool rap.
Okay, hi, thanks everyone for having us. My name is Max, this is Jordan, and Vince. And we are going to be, before I begin a discussion of what we did, I want to go over for some of the non-biologists in the room, basically what the underlying concepts of what we're trying to do. So there are these regions of DNA that you find all of the enhancers. These are non-coding regions. That means they don't actually turn into a protein, but they are extremely important for the regulation of individual genes. So it's a lot of biologists don't really consider them to be very sexy because it's really hard to study. You don't get a protein product you get to play with, and yeah, just for a lot of reasons. I'd like to change that and make them sexy top. So hopefully after this you'll think so. So they'll be about 100, 1,500 base pairs long, and basically what they do is they act as binding sites for proteins to come along, specifically transcription factors or other things that facilitate the transcription of genes downstream or upstream. So they'll have some sort of spatial relationship to a gene, but that is hugely variable. And it's really hard, they can be really hard to find and really hard to characterize for these reasons. and they maybe locate upstream or downstream of your target genes. So with this in mind, what we want to do is try to predict some of the functional effects of these rare SNPs that we're identifying in these enhancer regions. So it's interesting because a single nucleotide polymorphism in this case can have really dramatic effects for how a protein is going to bind there, how a transcription factor or some other protein binding or DNA binding protein that facilitates transcription will bind. So what we did was we used 1,000 genome projects looking for common SNPs and finding every single rare SNP that was in Onno's genome. So everything that did not pop up in 1,000 genomes project. Using these SNPs, we then ran them through this the deep sea convolutional neural network, which is this model that's been developed by this group at Princeton for for predicting what the effect of a single SNP will be on the binding affinity of different chromatin binding proteins. Okay, so just a little bit of the results. So we were able to identify 236 ultra-rare SNPs in Onno's genome. So these were SNPs that didn't pop up anywhere in the 1,000 genome project. Of those, running through the deep sea convolutional, the deep sea model, we were able to identify 35 SNPs with a functional significance score of less than 0.01. So that's pretty good. There's a lot to go with this stuff. We need to go further and make sure that we filtered everything out correctly. And it would be great. We didn't really have enough time. There's a lot of different things happening to compare against Onno's normal expression, although that may or may not have significance because it could be just underlying SNP in his genome may be affecting this in other ways. So there's a lot that can really be done with this. And I think it's really exciting the way that machine learning is changing the field of bioinformatics, especially being able to translate DNA into, so think of DNA as like sentences and words, being able to translate that, like find the grammar of DNA and be able to say, this is like identify the functional segments. So in the past, like, you're able to identify motifs, identify top box, things like that, these functional elements. But if we can use machine learning to identify larger functional elements and spatial regulation of these things, it'll be really awesome. So yeah, some of the things we need to do is really refine some of the machine learning, the deep learning algorithms to improve performance and extend utility. And this is just an overview of what we did. And that's our team.
I told you that was boring. Great job, guys. That was awesome. I mean, like, really, yeah, we don't know answers in my opinion as well. All right. A couple more teams. I know you guys are back there. I know. Onno knows too.
I just don't know which one. Protein. Wait.
Protein and
Mutation 0. Mutation 0.
Yep. Mutation 0. Mutation 0. Protein has to wait. Mutation 0. Welcome. By the way, one of their team members has the coolest GitHub name I've ever heard. I'll let you figure out who that is.
All right, so good evening, everyone. We are the scientists and engineer from the Mission Zero team. So I'm Shai. Calvin.
Hola.
All right, and we have also tapped 2 more members who joined us online. First of all, today we're going to present you how can you rank the classification using the combined annotation scoring matrix and also use the G-network approach. I think by now everyone now very convinced that we live in an era that we have a lot of genomics data, not for NF2 specific, but all data in general. Say we have 1,000 genomes, we have encode, and people have different way of how can we make use of this different data set here to predict which gene is important one. So people can use like concentration score, protein interaction, also maybe like functional element in the genome. But now our approach here, we try to find a way that we can integrate all the information here and try to actually make sense of the case at hand here. To be specific, we decided to use this model from this article in Nature from Jay Chong's team. These two here, basically they're called the combined antigen dependent depletion. The idea is that is to combine all the information from the ensemble variants, effect predictor, or ENCODE project, and also the UCSC genome browser. And then from that, they use the spot vector model to train on this dataset and then try to predict what is the effect from missing individual variants in the genome. The beauty of this tool is that it predicts not just on the gene level, but also at the base pair level of actually every base pair in the genome. You can predict what is the effect that it could be on both SNP and indel as well. So now let's see how can we make use of this geometry to solve this issue at hand. In this case, if we compare the definition on the we compare the genotype from the tumor brain and from blood, then we can figure out what are the somatic mutations that this tissue could have. And you can see here there are a lot of mutations here, and if we run even the pre-trained model on this dataset, it's going to take a lot of time. So instead of that, we also try to scope down to the set that is more specific to the NF2 gene. We use this tool here called the BioGrid, which can tell us what are the genes that are related to the NF2. So with that, we can boil down to smaller set, and then we can also remove the known variants from 1,000 genome entities as well. Finally, we come down into a set of 1,600 variants. These are the distribution of those variants along the chromosome 2, 3, 11, 16, and 22. We can do actually more because we have the patient data plus sibling as well, right? So if we subtract the variants that we found on the patient from variant in the sibling, we can actually come up with a set of rough idea of what could be potential germline mutation. Now there's a caveat here because the sibling not gonna have the same set of chromosomes because you can get different chromosomes from your father and from your mother. So there's gonna be a whole lot of false positive here. So now in this case, we scope only on the variants on the NF2 and EGFR, and so subtract a lot of them from the 1,000 genome entities as well. So this is what the result looks like. You can see here that on the x-axis that the position on the NF2 gene, and these are the somatic mutation and germline mutation that we predict. On the y-axis, those are the normalized score of the expected potential to have functional effect. You can see here that a lot of variants that cluster at the beginning of the gene here that could potentially have the effect, and also like somewhere near toward the end as well. Now for the germline here, we have to very careful on interpretation because this could be just the difference between Onno and his brother. It may not be actually the germline mutation. But in the conclusion, I would say that now I show you that we can possibly combine all these annotated information together and pass the this very unique data set here to prioritize what are the variants that could affect the NF2 patient. We usually have already run this CADD score on the whole genome, and we just get the result like half an hour ago. And I would think that if in the future, if we can genotype like parents, or we can get other NF2 patients, now we can use that to reduce the false positive that you found in the germline mutation, and really pinpoint to what are the overall landscape of both somatic and germline mutation, and then show which position in the gene that you actually should target on. Thank you.
Wow, that was awesome. Thank you. All right. All right, I think we have at least one more team left. Yes. So pro team. We'll find them. There we go. Come on up, guys. I would say that I spent more time with this team than any other team. So they had a lot of questions. They learned a lot. And I think they came up with something very good. So without further ado.
Thank you so much for helping us. And thank you again, Onno, for providing the data and putting this together. Our team name is Protein. Protein, it's supposed to be protein, but M because we just, I'm trying to be clever and funny this time. My name is Mary Chris.
Welcome. Jen, Brandon, Ronald.
And we're an interdisciplinary team where I'm the biologist, computer science, biomed, and chemical engineering. So let's go. We know based on literature that NF2 is located in the 22q chromosome, and it codes for Merlin protein. And the Merlin protein has, there's a difference between the closed conformation and open conformation. I basically just focus on two different papers, review papers. This one and this one came from one paper, and the light pink one is from one. So we found that from what's known out there is that the Merlin protein is phosphorylated at 3 different sites. And with this paper, it says a close Merlin conformation leads to activation of mitogen signaling pathway. Phosphorylated is inhibition. But for the other paper, it says the phosphorylation at these 2 sites, but when it binds to AKT, it inhibits binding of Merlin with PIKE-L, which inhibits PI3 kinase activity. But it promotes polyubiquitination and promotes degradation and all that. So with this in mind, we have a question. Well, how can we search for unique proteins from the genomic data? And what is the likelihood of these unique proteins can be phosphorylated? So I know that we have genetic data, right, from Onno's brother, which we consider as normal. And we have Onno's control normal, internal control, blood, and then we have the tumor sample from Onno. And
so the method that we wanted to find the unique proteins relevant to the tumor. And so what we did was we took the genes from well, we took the genes from all 3 genomic files, and we found, as you can see in the Venn diagrams, we found what was unique, specific to the tumor in comparison to Onno's blood and his brother's blood. And we studied those in terms of their probabilities for phosphorylation at different sites. So specifically, we take the VCF files, we ran them through the SNP-F tool and found the RS numbers. The RS numbers are reference SNP ID numbers. And we ran those through this eSearch script, which would give us the FASTA files, and using the FASTA files would give us the amino acid sequence. Using the amino acid sequence, we fed that into this MuseSite Deep program, which uses machine learning to predict what the probabilities are of a threonine or another amino acid, a tyrosine, being phosphorylated at a certain site. And so
because that was our ideally this is how you would want to analyze this data. You want to individually look at the RS numbers. However, we are provided with 2 files. One file has Onno's blood, and the other file contains Onno's brother's blood and Onno's tumor VCF file. And so it's hard for us to do the other way of analyzing. So what we did was we just we obtained the RS numbers from Onno's blood, and we obtained the unique RS numbers. So we basically just compared the unique RS numbers from that one file that contains 2 samples. And from that, we found that there are 149. We compared the RS, we say RSS for the blood, and then the RSY is the unique tumor RS numbers. And from that, we found 149 unique RSZ. We just say that this is the one that we're really interested in, and we fed that through the machine learning program, and we found 218 proteins. So our result shows that we have, from the 149 unique RS numbers, 44 gave us no result through NCBI. And we found 105 results from NCBI. And this is the one that corresponds to 218 proteins. And for further studies, here, if we set the cutoff, so this is the frequency of proteins that have likelihood of phosphorylation site. So if we set the cutoff to 50, we find that there are 67 proteins. If we set the cutoff to, I have a cheat sheet. If we set the cutoff to 100, we have 29 proteins. And if we set the cutoff to 200, we have 7 proteins. So further studies for this is this is what we've accomplished this weekend. Further studies. So what we can do now is we can provide you this unique proteins that have high likelihood of phosphorylation site. And this is the very essence of personalized medicine. So ideally, like for normal biologists, you take from the rat samples, right, and then put it to human. But here we have something that he gave us his sample, and we just deciphered that data, and we actually found 67 proteins if we set the cutoff to 50. And we can look that further and see and do like immunohistochemistry studies, confirm if they are even present in other tumor samples, existing tumor samples that we have in the lab that maybe UCLA or I know UCSF has, if they have NF2 patients that are formalin fixed tissues or frozen tissues, they can cut that off and do imaging studies just to see whether or not these proteins are even present. So that's it.
Thank you. And a marathon presentation as well. If you guys can go get a picture taken. Is there anybody in this room that wants to present and has not yet presented? Oh yeah, right, yes. Hi guys. Come on up. What's the name of your team again? T-Rex? TRS. They stayed with TRS. They told me they were going to change the name. It's going to be great. Here, come on. TRS.
So we are Tiffany, Irina, and Chu, astonishingly called Team TRS. And so collectively, we have experience in machine learning, software engineering, and biochemistry in academia and biotech. So we're in the general vicinity of doing data analysis for genomic sequences, but we have not ourselves ever done it before. So we took this as an opportunity to do that by taking on the ranking. So our sequence analysis pipeline was to take the VCF files, feed them through FK tool sets, extract samples. Since we were limited on time, we decided to focus on chromosome 22. Took differences between Onno's tumor and his normal, as well as information from his sibling. We annotated them using NFR, and then focused really on the exonic regions and excluded the synonymous. So one question that we had going in, kind of based on what Marco's presentation was, is were Onno's variants somatic or germline? And so based on the data that we were given, we didn't see any SNPs or indels in Onno's blood or his siblings' blood samples, the normal samples. So we understand now that they actually are somatic mutations. And he does not carry any of them. And so biologically, so what we discovered were two mutations, single nuclear polymorphisms, one that affects exon 3 and one that affects exon 6. One has already been annotated in ClinVar, but the C to G was actually a novel discovery, and both of them are actually stop gating mutations.
So we also look at all the variants of chromosome 22. So we summarized total variants, exonic variants, and non-synonymous exonic variants for the 3 samples. And we see that there are more variants in the tumor sample than in blood normal or sibling normal. And to narrow down our list, we also compared between tumor versus normal, tumor versus sibling, and normal versus sibling. So we found there are 7 different non-synonymous exonic variants between tumor and normal blood, corresponding to 5 genes, and there are 93 different non-synonymous exonic variants between tumor and sibling blood, corresponding to 38 genes. So we're going to focus on the 7 variants and the 38 genes. So here are the 7 non-synonymous exonic variants that are present in tumor but are not present in blood normal. So we see here the 2 stop gain mutations in NF2. And for the other 5 variants, the functions of those genes are not directly implicated in cancer. Here are the 38 genes that are present with variants that are present in tumor but not in his sibling. And we also did functional annotation for the 38 genes, and we found a few interesting genes. So for example, this COMP gene is associated with neurotransmitter function. And also MAPK is a well-known gene implicated in cancer. So it's an essential component in the MAPK signaling pathway. Another interesting gene we found is this MN1 gene. So this variant is a 3 base pair insertion. It's present in almost tumor and blood samples, but not in the sequence data. And the function of this gene is proto-oncogene in Genomic. So in conclusion, we identified 2 stop gains in the NF2 gene, and also we discovered a novel variant in MN1. So thank you very much.
I'd say that's a pretty clear result. Great job. Cool. Yeah. Awesome. Did we miss anybody? Just want to triple check. Anybody want to present who hasn't presented? Yes. Wow, yes, that was an enormous amount of work.
There's more people.
What?
There's more people.
There are a couple of people. Yes, there's a couple of teams who haven't presented, but I think if they don't want to present, we will let them. We will let them not present. Are you sure? You sure? Okay, gotcha. So that's it. Wow, that was an enormous amount of information. So here's what we're going to do now. What I would like everybody to do is let the judges get a beer and then come back here. We'll have a judge discussion because not everybody puts stuff on the Google Doc, particularly me and Onno. And we'll talk about 3 teams, and we'll let everybody else go to happy hour. And then in about 10 minutes, we'll announce sort of the results of our finding. But regardless of like sort of the winners and so on and so forth, I just want to say the amount of science that was done in the last, 2.5 days is, yeah, like 48 hours. Absolutely incredible. So here's to all of you. Amazing job. The only other thing I would ask is just let the judges through the beer line, and then we will have a meeting, and then we will announce what's going on. So thank you to all of you. You guys are amazing.
Yes. Oh, great. What is your yeah, it's not that good. Yeah. So that's why we're very careful. That's why. Judge. I'm a medicinal chemist. And I work for the which is the largest organization that supports research for so I'm director of research.
I came late.
Thank you for coming.
So yeah. Yeah, I mean, I haven't written anything down.
I have 2 assistants.
That sounds totally reasonable. Yeah. Yeah.
I know, it's like on a break.